Welcome to the Cosmic Guide to Sleep. Tonight we'll enter the hidden world of fungi. Fungi are among Earth's most overlooked, yet ubiquitous life forms. As you stroll through the forest, a vast fungal network may lie hidden beneath your feet. These organisms rarely draw attention, for they neither move nor make a sound. Yet fungi play an indispensable role. They are nature's most efficient recyclers, responsible for breaking down dead organic matter. Without fungi, forests would be buried under layers of fallen leaves and trees. Nutrients couldn't be recycled, and soil would gradually become depleted. Through decomposition, fungi release elements like carbon, nitrogen, and phosphorus back into the environment. These elements are essential nutrients for plant growth. Fungi act as the digestive system of ecosystems, transforming waste into resources. They form close partnerships with trees and other plants. This relationship is known as mycorrhizal symbiosis. Fungi help plants absorb water and minerals. In return, plants provide sugars produced through photosynthesis. This mutually beneficial relationship has persisted for hundreds of millions of years. Fungi also aid in forming stable soil aggregates. Their mycelia bind soil particles together like microscopic cables. This not only prevents soil erosion but also enhances the soil's water-holding capacity. During droughts, this structure is vital for plant survival. Some fungi can break down pollutants, purifying soil and water sources. These fungi can degrade pesticides, industrial chemicals, and even plastics. This ability, known as bioremediation, is a crucial tool for environmental protection. Fungi also serve as a food source for many animals. Insects, birds, and small mammals all feed on mushrooms. Simultaneously, fungi aid these animals in digesting food. Within gut microbiomes, fungi play vital roles. Fungi even influence the atmosphere and weather. The spores they release can act as cloud condensation nuclei, promoting cloud formation and precipitation. This demonstrates how fungi connect the underground and the sky. Despite their immense importance, fungi remain largely unseen. They work silently beneath the surface, needing no recognition. This quiet existence is quintessentially fungal. Understanding these hidden organisms can transform our view of nature. The next time you see mushrooms on the forest floor, remember they are merely the tip of a vast fungal network. Beneath lies a world teeming with life and vitality. Fungal networks connect trees and facilitate nutrient sharing. Scientists refer to this network as the Wood Wide Web. This metaphor vividly describes fungi's role in transmitting information across ecosystems. Mature trees can nourish saplings through fungal networks. This intergenerational support enhances the forest's overall resilience. When a tree faces insect attack, it sends warnings through the fungal web. Nearby trees then activate their defenses in advance. This collective response is truly remarkable. Fungal networks also moderate competition among plants. By distributing resources, they prevent any single species from dominating. This promotes biodiversity and stabilizes ecosystems. Fungal networks act as a buffer against climate change. They help plants cope with drought, heat, and extreme weather. Mycelium networks extend into areas beyond plant root reach, supplying deeper moisture and minerals to plants. This support system is especially vital under harsh conditions. Fungal decomposition regulates the global carbon cycle. As they break down organic matter, some carbon is stored in soil. This helps reduce atmospheric carbon dioxide levels. Soil carbon pools rank among Earth's largest carbon reservoirs. Fungi make substantial contributions in this regard. Some fungi can even directly capture atmospheric carbon. Through specialized metabolic pathways, They convert carbon dioxide into organic carbon. This capability holds potential applications for climate change mitigation. Fungi also serve as vital sources for medicinal treasures. Numerous antibiotics, immunosuppressants, and anti-cancer drugs originate from fungi. The discovery of penicillin has saved countless lives. This is just one example of fungal chemical diversity. Fungi's applications in agriculture are equally significant. They serve as natural biocontrol agents, reducing chemical pesticide use and protecting ecosystems. Fungal fertilizers enhance crop yields and improve soil health. Throughout human history, fungi have been constant companions. From bread fermentation to alcoholic beverage production, from cheese ripening to soy sauce and miso brewing, fungi enrich our culinary culture. Though fungi seldom take center stage, they are indeed the silent architects of our world. Through quiet persistence and patient labor, fungi sustain the cycle of life on Earth. When we learn to appreciate these hidden lives, we gain deeper understanding of nature's complexity and wisdom. Fungi teach us humility, reminding us to notice the unassuming. For sometimes the most vital forces lie hidden in the quietest places. Fungi are a highly distinctive class of eukaryotes, neither plant nor animal. They constitute a separate biological kingdom. Members of the fungal kingdom share several fundamental characteristics. The most distinctive feature is their cell walls composed of chitin. This resilient substance is also found in insect exoskeletons. This fundamentally differs from the cellulose cell walls of plants. The fungal body structure is primarily composed of hyphae. Hyphae are slender, tubular cellular structures. They continuously branch and intertwine to form a mycelium. The mycelium is the primary form during the fungal nutritional growth phase. This network structure enables efficient exploration of the surrounding environment. Fungi grow by extending the tips of their hyphae. This growth pattern allows them to penetrate various substrates, including soil, wood, and even tiny crevices in rock. Fungi also possess a highly unique nutritional strategy. They obtain nutrients through external digestion. Fungi secrete powerful enzymes into their surroundings. These enzymes break down complex organic compounds, such as lignin, cellulose, and proteins. They then absorb and utilize the resulting small molecules. This feeding method differs from both animals and plants. Fungal reproduction is equally distinctive. They primarily reproduce by producing spores. Spores are the reproductive units of fungi. Extremely small, they can be dispersed through air, water currents, or animals. Some fungi produce fruiting bodies, commonly known as mushrooms. These structures are specialized for spore production and dispersal. The diversity within the fungal kingdom is astonishing. Scientists have described over 150,000 fungal species, though the actual number may reach millions. Fungi can be grouped into several major phyla. The Basidiomycota includes familiar mushrooms, characterized by producing basidiospores on gills. The Ascomycota encompasses yeasts and molds. Their spores form within asci. The Zygomycota contain common bread molds. The Chytridiomycota primarily inhabit aquatic environments. More primitive fungal groups also exist. Yeasts represent a unique class of single-celled fungi. They reproduce through budding or fission. They play vital roles in food industries and scientific research. Molds form characteristic filamentous colonies. They rapidly spread across organic surfaces. They are crucial in natural decomposition processes. Lichens represent a unique symbiotic association, composed of fungi and algae or cyanobacteria. This symbiosis enables them to thrive in extreme environments. The classification system for fungi is continually evolving. Advances in molecular biology techniques have provided scientists with new insights into fungal relationships. For instance, microsporidia were discovered to be a specialized type of fungus. Several groups have been reclassified and reorganized. Understanding fungal taxonomy aids in recognizing their evolutionary history and better utilize these biological resources. Fungi exhibit remarkable size variation. Some yeast cells measure only a few micrometers, while subterranean fungal networks can span thousands of hectares. The largest known fungal organism resides in Oregon, USA, with mycelium extending over 8.9 square kilometers, and estimated to have grown for millennia. Fungal lifespans also vary greatly. Some species survive only days, while fungal colonies may persist for tens of thousands of years. This diversity enables fungi to adapt to virtually any environment, from polar regions to deserts, from high mountains to the deep sea. Fungi are found in nearly every corner of the earth. Consequently, their ecological roles vary greatly. Some act as decomposers, others as symbionts, while still others function as pathogens or parasites. This multifunctionality makes fungi indispensable components of ecosystems. Studying fungal characteristics and classification not only satisfies scientific curiosity, but also deepens our understanding of life's diversity, while uncovering new applications in medicine, agriculture, and environmental protection. Though fungi sometimes resemble plants in appearance, they are actually more closely related to animals, a connection confirmed by molecular biology research. Both fungi and animals evolved from a common ancestor. This ancestor was a single-celled flagellate organism that diverged from plants approximately 1 billion years ago. Fungi exhibit distinct cellular structures compared to plants and animals. The primary component of fungal cell walls is chitin, the same material forming the exoskeletons of insects. Plant cell walls, in contrast, are primarily composed of cellulose, while animal cells lack cell walls altogether. These differences reflect their divergent evolutionary paths. Nutritional mode represents another key distinguishing feature. Plants are autotrophs capable of photosynthesis. They utilize solar energy to convert carbon dioxide into organic matter. Fungi and animals are both heterotrophs requiring external nutrient sources. Yet their acquisition methods differ entirely. Animals ingest food for internal digestion. Fungi absorb nutrients through external digestion. This divergence leads to fundamentally different bodily structures. Each group also exhibits unique energy storage mechanisms. Plants store energy as starch. Animals primarily use glycogen as their energy reserve, while fungi utilize both glycogen and trehalose. These biochemical differences reflect distinct metabolic strategies. Mobility represents another clear distinction. Most animals possess active locomotion capabilities. Plants and fungi generally lack autonomous movement. However, fungal hyphal growth can be considered a specialized form of movement. They sense their environment and grow toward nutrient sources. This growth movement, though slow, is remarkably precise. Reproductive strategies also exhibit significant variation. Animals typically produce offspring through sexual reproduction. Plants possess complex systems of both sexual and asexual reproduction. Fungi exhibit the most diverse reproductive methods. They can reproduce both sexually and asexually, and produce various types of spores. This flexibility allows fungi to rapidly adapt to environmental changes. Distinct cellular differences are also evident among the three. Plant cells contain chloroplasts and large vacuoles. Animal cells possess centrioles but lack cell walls. Fungal cells have cell walls but lack chloroplasts. These organelle differences dictate their fundamental lifestyles. Genetic research reveals deeper distinctions. Fungal genomes exhibit unique characteristics. Certain metabolic pathways show greater similarity to animals, while diverging significantly from plants. For instance, in cholesterol synthesis, fungi and animals utilize comparable biochemical pathways, whereas plants employ entirely distinct methods. The nervous system is a feature unique to animals. Neither plants nor fungi possess nervous tissue. However, recent research indicates fungal hyphal networks can transmit electrical signals. This form of communication exhibits certain intelligent characteristics. Although distinct from the animal nervous system, it demonstrates complex biological communication capabilities. Growth patterns also reveal fundamental differences. Animal growth is deterministic and limited. Plant growth is open-ended yet follows fixed patterns. Fungal growth is entirely open and adaptive. Mycelial networks can adjust growth directions based on environmental conditions. This flexibility enables fungi to efficiently explore resources. Their strategies for coping with environmental stress differ significantly. Animals can avoid adverse conditions through behavior. Plants adapt to changes through physiological adjustments. Fungi, however, can enter dormancy to await favorable conditions. Their spores can survive for decades or longer. This resilience allows fungi to endure harsh periods. Their ecological roles also show distinct differences. Plants primarily function as producers, establishing the energy foundation. Animals primarily act as consumers, transferring energy. Fungi primarily serve as decomposers, completing material cycles. This division of labor maintains ecosystem balance. Understanding these distinctions holds not only academic significance, but also aids our comprehension of nature's operational principles. Such knowledge proves particularly vital in biotechnology applications. For instance, developing new drugs can target fungal-specific pathways. In agricultural production, symbiotic relationships between fungi and plants can be harnessed. In environmental protection, fungi's decomposition capabilities can be harnessed. Fungi, plants, and animals, each kingdom possesses distinct characteristics. Together, they form the rich diversity of life on Earth. Every kingdom embodies unique survival wisdom, deserving our respect and in-depth study. Hyphae are the fundamental units of fungal life. They function as the roots, stems, and leaves of the fungal world, yet possess far greater structural flexibility than plant organs. A single hypha is a slender tubular cell, typically measuring just a few to several dozen micrometers in diameter, and nearly invisible to the naked eye. These minute structures wield astonishing power. The hyphal tip represents the most active growth zone, where new cell wall material is continuously synthesized, propelling the hypha forward through its substrate. Hyphal growth rates vary significantly by species. Some extend only a few millimeters daily. Others can reach several centimeters under favorable conditions. This growth appears slow yet unwavering, as hyphae continuously branch and intertwine. They form a network structure known as the mycelium. The mycelium is the true nutritional body of fungi. It can extend several meters within soil. Some even cover entire forests. The mycelium exhibits diverse morphologies. Some form loose networks, while others densely intertwine into hyphal strands. Still others specialize into distinct organs for nutrient uptake. The hyphal body serves multiple vital functions. Primarily, it acts as the primary organ for nutrient absorption. Mycelia contact the environment through their vast surface area, secreting digestive enzymes and absorbing the resulting breakdown products. This external digestion strategy proves highly efficient. Secondarily, it functions as a crucial pathway for material transport. Active cytoplasmic streaming occurs within the hyphae, enabling the transport of nutrients from enriched areas to where they are needed. This transport requires no heart or pump pressure, relying solely on osmotic pressure and cytoplasmic streaming. Mycelia also function as environmental sensors. Mycelial tips detect temperature, humidity, and chemical gradients, adjusting growth direction accordingly. This ability allows fungi to precisely locate resources. Mycelial networks exhibit remarkable adaptability, automatically altering direction when encountering obstacles, accelerating branching upon discovering abundant nutrients, entering dormancy under unfavorable conditions. This intelligent growth pattern is astonishing. In certain fungi, hyphae specialize into haustoria. These specialized structures penetrate plant cell walls to establish tighter nutritional connections with the host, without causing immediate cell death. Other hyphae form sclerotia, dormant structures formed by tightly intertwined hyphae that help fungi survive harsh environmental conditions before resuming growth when conditions improve. Fusion between hyphae is also common. When two compatible mycelia meet, they break through septa to achieve cytoplasmic connection. This fusion makes fungal networks more complex, allowing nutrients and information to flow over greater distances. The lifespan of mycelia varies greatly. Some survive only a few weeks, while others persist for millennia. The oldest mycelium discovered to date has been growing underground for 2,400 years. The scale of mycelia is also astonishing. The largest mycelium network, found in Oregon, USA, spans 8.9 square kilometers, equivalent to 1,600 football fields. Mycelium plays an indispensable role in ecosystems. As nature's primary decomposers, they break down stubborn substances like lignin and cellulose, releasing organic nutrients back into the environment. Mycelium also forms vast underground networks, connecting plant root systems, facilitating the exchange of nutrients, water, and information. This network is known as the mycorrhizal network. Recent research reveals that mycelium can also conduct electricity. These electrical signals may serve for information transmission. Though distinct from animal neural signals, they demonstrate sophisticated biological communication capabilities. These properties of mycelium inspire technological innovation, from self-healing materials to intelligent network design, from environmental remediation to novel computational models. Fungal mycelium continues to provide us with inspiration. Mushrooms are the most captivating entities in the fungal world. Yet they are merely the reproductive structures of fungi. As transient as the fruit on a tree, the mushroom's life cycle begins with primordia formation. These minute bud-like structures develop underground, requiring optimal temperature and humidity to mature. The primordia already contain the complete blueprint of the future mushroom. When environmental conditions align, primordia begin to swell. Mycelium channels vast quantities of water and nutrients to this site, propelling the mushroom through the soil. This process typically unfolds at night or dawn, when heightened humidity facilitates cellular expansion. Some mushrooms fully emerge within hours. This rapid growth appears miraculous, yet it achieves its scale through cellular enlargement rather than division. Mycelium pumps water into the developing mushroom, causing cells to expand rapidly like inflating balloons. The mushroom's morphological structure is highly specialized. The cap protects the underlying hymenium. The stem elevates the mushroom to an optimal height. Gills or tubes serve as spore-producing surfaces. Some species also feature structures like rings or basidiophores. These adaptations enable efficient spore dispersal. The mushroom's maturation process is meticulously precise. First, the cap gradually flattens from a hemispherical shape to maximize space for spore release. Then, the hymenium begins producing spores. These minute reproductive units are released at specific times. Spore production is astonishing. A typical mushroom releases millions of spores daily, and billions over its entire lifespan. This numerical advantage ensures reproductive success. The timing of spore release is meticulously regulated. Most mushrooms choose to release spores when air conditions are stable, typically during early morning or evening hours. This allows spores to disperse more effectively with the wind. Some mushrooms employ an active ejection mechanism, propelling spores through subtle structural changes. Others rely on rain impact to aid dispersal. The lifespan of mushrooms is extremely brief. Some species survive only a few hours. Most complete their mission within a week. Before initiating decay and decomposition, the mushroom's aging process is equally orderly. First, edges curl and discolor. Then texture softens and gradually liquefies, finally returning to the earth as nutrients. Yet the significance of this fleeting existence is profound. Mushrooms ensure the dissemination of fungal genes, enabling populations to propagate across broader territories, while simultaneously providing sustenance for other organisms. Different mushrooms adopt distinct survival strategies. Some emerge en masse after rainfall, exploiting high humidity to rapidly complete reproduction. Others appear sporadically throughout the year, employing a more resilient reproductive strategy. Certain mushrooms even possess the ability to glow. This bioluminescence may attract insects, aiding in dispersing spores to distant locations. Mushrooms also engage in close interactions with animals. Numerous insects lay eggs and reproduce within mushrooms. Larvae feed on fungal tissue, while simultaneously aiding in spore dispersal. Mammals like squirrels also forage for mushrooms by hanging them on tree branches to dry. This storage behavior unexpectedly aids spore dispersal. The relationship between humans and mushrooms is equally ancient. Our history of foraging and consuming mushrooms spans millennia. Today, mushroom cultivation has become a significant industry, producing millions of tons of edible fungi annually. Understanding the mushroom life cycle is not only fascinating, but also holds significant practical value. In conservation, it helps protect rare species. In cultivation, it guides us to increase yields. In medicine, it inspires the development of new drugs. Next time you see mushrooms in the forest, remember this is a miracle of life, a masterpiece of evolution spanning millions of years. Though fleeting, it radiates the brilliance of life's wisdom. Spores are the seeds of the fungal world, yet far smaller than plant seeds. They serve as the fundamental units of fungal reproduction, bearing the vital responsibility of perpetuating life. A single spore typically measures only a few micrometers, requiring a microscope to discern its form. But within these minuscule structures lies astonishing life force.
Spore shapes vary endlessly.
Some are spherical, others elliptical. Some bear intricate decorative patterns, while others grow with specialized appendages. These morphological traits are not random. Each represents an ingenious design forged through long-term evolution. They enable efficient dispersal across diverse environments. The process of spore production is a natural marvel. Within the fruiting body's gills of a mushroom, Each basidiospore cell generates 4 spores. These spores are ejected explosively, achieving initial velocities of tens of kilometers per hour. That such minuscule objects can attain such speeds is truly awe-inspiring. The timing of spore release is precisely calculated. Most fungi release spores during dry air conditions, as arid environments facilitate their dispersal. Certain species release spores after rainfall, to leverage moist conditions protecting fragile spores. Others release spores at specific temperatures, ensuring propagation under optimal conditions. Wind serves as the primary dispersal agent for spores. Tiny spores can ride air currents to altitudes of thousands of meters, undertaking long-distance journeys. Some spores can even cross oceans to spread between continents. This formidable dispersal capability enables fungi to inhabit every corner of the earth. The quantity of spores in the atmosphere is staggering. Thousands may exist per cubic meter of air, with even higher concentrations in forested areas. These microscopic life forms constitute the airborne zooplankton community. Though invisible to the naked eye, they form a vital component of ecosystems. Rainwater also plays a vital role. When raindrops strike mushroom surfaces, they generate subtle vibrations that shake spores into the air. Some fungi's spore capsules rupture under raindrop impact, releasing their contents. Though this method covers shorter distances, proves highly effective for local dispersal. Animals serve as another key dispersal agent. Insects pick up spores while foraging, carrying these tiny life forms to new locations. Birds and mammals transport spores over greater distances by consuming mushrooms. Some spores even survive passage through animal digestive tracts to reach new environments while retaining viability. Certain fungi have developed specialized strategies. For instance, puffball fungi emit foul odors to attract carrion-feeding insects like flies. The spores then adhere to the insect's exoskeletons and are dispersed as the insects fly. This intricate symbiotic relationship demonstrates nature's remarkable ingenuity. Waterborne dispersal is common among aquatic fungi. These fungi produce motile spores with flagella that can freely swim in water seeking suitable environments. Upon finding a suitable host or substrate, they attach and begin development. Though limited in range, this dispersal method proves highly effective within aquatic ecosystems. The fate of spores after landing varies greatly. Most spores never germinate. They may land on unsuitable surfaces, or be consumed by other microorganisms. Only a fortunate few find ideal conditions. This enormous attrition rate explains why fungi produce such vast quantities of spores. When a spore encounters a suitable environment, the miraculous germination process begins. First, the spore absorbs moisture from its surroundings, gradually swelling in size. This stage demands precise humidity control. Excessive dryness prevents activation, while excessive moisture risks decay. After hydration, the spore initiates metabolic activity. Stored nutrients begin converting to fuel the germination process. Enzymes soften the spore wall, preparing for the emergence of the germination tube. This process requires suitable temperature, varying among different fungal species. The germination tube extends from the spore. The first glimmer of new life. It begins exploring its surroundings, seeking nutrient sources and growth direction. If environmental conditions remain favorable, the germination tube continues elongating, forming new hyphae. Newly formed hyphae are extremely fragile and must quickly locate nutrients. If usable organic matter is present nearby, the hyphae will grow toward it, beginning to establish their territory. This stage is crucial for the fungus's survival. Some spores require specific conditions to germinate, such as passing through an animal's digestive tract, or enduring a period of low temperatures. These requirements ensure spores germinate only at the optimal time, avoiding energy waste under unfavorable conditions. This is an intricate mechanism shaped by long-term evolution. The success rate of spore germination is extremely low, possibly as low as 1 in 10,000. Yet, given the vast quantities of spores produced by fungi, this ratio suffices to sustain populations. A medium-sized mushroom can release billions of spores. Even if only a tiny fraction succeed, it ensures the continuation of the lineage. Environmental factors significantly influence germination. Temperature must fall within a specific range. pH levels must be suitable. Oxygen content is equally critical. Some spores additionally require specific signaling molecules. These intricate regulatory mechanisms ensure spores germinate only at the optimal moment. In agricultural production, understanding spore germination characteristics is vital for controlling crop diseases. By disrupting the germination conditions of pathogenic spores, the use of chemical pesticides can be reduced. In edible mushroom cultivation, mastering spore germination patterns enhances cultivation success rates. Spores possess remarkable durability. Some spores can survive for decades, entering dormancy under extreme conditions, to await the day when the environment improves. This trait enables fungi to endure harsh periods and rapidly revive when conditions improve. Studying spore dispersal and germination holds significant importance for understanding ecosystems. Fungi connect different habitats through spore dispersal, facilitating material cycling and energy flow. They also play a role in ecological restoration by introducing beneficial fungi through spores to aid damaged ecosystems in regaining health. A spore's journey is fraught with challenges. From release to landing to germination, each stage a test of survival. Yet this harsh selection process forges fungi's formidable adaptability, enabling them to thrive in diverse environments. Next time you see a mushroom, consider those microscopic spores, embarking on their grand journey. This invisible dust of life quietly reshapes the world. Though tiny, it holds infinite potential. When a great tree falls in the forest, what seems like life's end unfolds another vital chapter. Fungi silently begin their work, transforming death into new life. This process, called decomposition, is one of fungi's most vital roles in ecosystems. Decomposition begins at the smallest scale. Fungal hyphae, like nature's scouts, are the first to reach dead organic matter. They secrete specific enzymes, precise molecular tools, capable of breaking down the chemical structure of plant cell walls, especially the stubborn molecules lignin and cellulose. Lignin is the most resistant component of plant cell walls. It imparts strength and durability to wood. Most organisms cannot break down this complex polymer, but white rot fungi possess this unique ability. They produce peroxidases and lactases, capable of gradually dismantling lignin's intricate structure. This process is slow yet relentless. At different stages of decomposition, various fungal species take turns. First, fungi capable of utilizing simple sugars rapidly colonize easily degradable sections. Next, species specialized in breaking down cellulose and hemicellulose take over. Finally, the experts on lignin enter the scene. This ordered succession ensures efficient decomposition. Fungal decomposition extends beyond forests. On grasslands, they break down dead grass and animal dung. In farmlands, they decompose crop residues. Even in urban parks, they quietly process fallen leaves and dead branches. These seemingly mundane tasks actually sustain the entire ecosystem's functioning. During decomposition, nutrients are gradually released. Essential elements like nitrogen, phosphorus, and potassium are liberated from organic matter into forms plants can absorb. Without fungal decomposition, these nutrients would remain locked within dead tissue, disrupting the ecosystem's nutrient cycle. During decomposition, fungi also create specialized microenvironments, where bacteria and other microorganisms gather around mycelium,
Forming à décomposer alliance.
each excelling at distinct decomposition tasks. This collaboration significantly enhances decomposition efficiency, while promoting biodiversity maintenance. Fungal decomposition is particularly crucial in deep soil layers, where they reach areas beyond plant root systems, converting deep organic matter into nutrients. These nutrients are transported through fungal networks and ultimately delivered to plant roots, completing the perfect cycle from death to new life. Decomposition rates are influenced by multiple factors, with temperature being a key regulator. Decomposition proceeds faster in warmer environments, while humidity is equally critical. Excessive dryness inhibits fungal activity, and excessive moisture may create anaerobic conditions, impairing the efficiency of aerobic fungi. Decomposition rates vary across ecosystems, with tropical rainforests exhibiting the fastest rates. High temperature, high humidity environments promote fungal growth, where organic matter decomposes completely within weeks, while in cold northern forests, the process may take several years. This variation shapes distinct ecosystem characteristics. Fungi also influence soil structure during decomposition. Mycelia act as natural binders, agglomerating soil particles into stable crumb structures. These structures retain moisture while allowing air circulation, creating ideal conditions for plant growth. The humus produced through decomposition is the essence of soil. This dark organic matter is nutrient-rich, enhancing soil fertility, improving water retention, and promoting healthy plant growth. Fungi are the primary drivers of humus formation. While decomposing dead matter, fungi also grow themselves. They convert some carbon and nutrients into their own biomass. These stored nutrients re-enter the cycle after the fungi die, forming a nutrient reservoir within the ecosystem. Fungal decomposition also regulates climate by controlling organic matter decomposition rates and influencing atmospheric carbon dioxide concentrations. During decomposition, some carbon becomes sequestered in soil, forming stable organic carbon reservoirs. This plays a significant role in mitigating global climate change. Certain fungi possess specialized decomposition capabilities, such as degrading pollutants, petroleum hydrocarbons, pesticides, and even plastics. These fungi demonstrate potential in bioremediation, cleaning contaminated land, restore ecosystem health. In aquatic ecosystems, fungi also serve as decomposers, breaking down fallen branches and leaves that enter the water, providing foundational nutrients for aquatic food webs. Certain aquatic fungi can even decompose water pollutants, helping maintain water body cleanliness. The decomposition process also impacts other organisms. Many insects feed on decomposing organic matter, forming complex relationships with fungi. Some insects aid fungal dispersal, while others feed on the fungi themselves. These interactions enrich ecological networks. Fungal decomposition even influences landscape evolution. By breaking down organic matter on rock surfaces, they accelerate weathering processes and promote soil formation. This seemingly minor role, accumulating over vast timescales, can transform entire landscapes. Understanding fungal decomposition holds implications for human activities. In agriculture, we can harness fungi to accelerate organic matter breakdown, enhancing soil fertility and reducing chemical fertilizer use. In waste management, fungi aid in processing organic waste to achieve resource recycling. In forest management, preserving fungal diversity is crucial, as they sustain forest health and regeneration. By facilitating nutrient cycling and supporting the growth of new tree generations. Without fungi's silent labor, forests could not maintain their vitality. As we stroll through the forest, the decomposition of every fallen leaf beneath our feet depends entirely on fungi's diligent labor. They serve as nature's recycling engineers, transforming death into life, connecting endings to new beginnings. This eternal cycle forms the very foundation of ecosystem sustainability. Though fungi's decomposing work may seem unremarkable, it represents one of Earth's most profound transformative processes. With patience and persistence, they sustain the cycle of life, reminding us that in nature, endings often herald new beginnings. Beneath the forest's tranquil surface lies a bustling subterranean world, where astonishing symbiosis thrives. Fungi and plants intertwine through mycorrhizal networks. This relationship, forged over 400 million years, stands as nature's most successful partnership. Mycorrhizae are the union of fungal hyphae and plant roots. The term originates from Greek, meaning fungus root, vividly describing this unique structure. When fungal hyphae encounter plant fine roots, they form intricate connection structures, neither parasitic nor simple cohabitation, but a true mutualistic symbiosis. Mycorrhizae are primarily classified into two types, epiphytic mycorrhizae and endophytic mycorrhizae. Epiphytic mycorrhizae are common in temperate forests. Fungal hyphae form dense sheaths on root surfaces, while extending inward into the intercellular spaces of root cortex cells, enveloping fine root tips like gloves. Pines, oaks, and beeches frequently form symbiosis with such fungi. Endomycorrhizae are more widespread across plant species, particularly with arbuscular mycorrhizal fungi, whose hyphae penetrate root cell walls, forming intricate branching structures, without compromising cellular integrity. This intimate contact dramatically enhances nutrient exchange efficiency. Approximately 80% of terrestrial plants form this symbiosis. The functionality of mycorrhizal networks is astonishing. Through minuscule hyphae, fungi explore microdomains in soil inaccessible to plant roots. They locate and absorb sparse nutrients, especially phosphorus, an element with poor mobility, along with trace elements like zinc and copper. These are essential nutrients for plant growth. In return, plants supply the carbohydrates fungi require. These sugars originate from photosynthesis and are precisely allocated to fungal partners through the root system. Research indicates plants transfer 10 to 30% of their photosynthetic products to symbiotic mycorrhizal fungi. This energy investment proves highly worthwhile for plants. The magic of mycorrhizal networks extends beyond nutrient exchange. They also transmit information between plants. When a plant is attacked by pests, it sends chemical signals through the mycorrhizal network, to alert neighboring plants to activate defenses early. These plants may strengthen cell walls or produce insect-repelling secondary metabolites. This early warning system boosts the survival chances of the entire community. Water is also shared through mycorrhizal networks. During droughts, trees with access to deep water sources can aid shallow-rooted neighbors via fungal networks. Mycelia act like microscopic pipelines transporting moisture. This mutualistic mechanism makes plant communities more resilient to climate change challenges, demonstrating remarkable collective intelligence. Mycorrhizal networks also influence forest regeneration. Seedlings connected to the network receive nutritional support, significantly boosting survival rates. Studies show that seedlings connected to mycorrhizal networks grow several times faster than isolated seedlings. This intergenerational support embodies nature's continuity. The scale of mycorrhizal networks in forests is astonishing. A single teaspoon of forest soil may contain kilometers of mycelium, connecting dozens of different plant species. These networks function like an underground internet, enabling rapid circulation of resources and information. Scientists refer to them as the Wood Wide Web. Different tree species play distinct roles within the network. Certain large trees serve as network hubs, with extensive root systems and robust photosynthetic capacity, capable of supporting numerous surrounding plants. These mother trees hold critical ecological significance. In forest ecosystems, protecting them safeguards the entire forest network. Mycorrhizal networks also facilitate chemical communication among plants, beyond warning of insect threats, to transmitting pathogen invasion signals, and even coordinating flowering times. This intricate communication system enables plant communities to exhibit collective behavior. Seasonal shifts influence mycorrhizal network activity. Spring's vigorous plant growth triggers peak network exchange activity, while winter induces relative dormancy. This rhythm synchronizes with ecosystem energy flows, demonstrating nature's intricate regulation. Mycorrhizal networks prove vital for ecosystem restoration, introducing suitable mycorrhizal fungi to degraded lands significantly accelerates vegetation recovery. Fungi aid newly planted species in environmental adaptation by supplying essential nutrients and moisture. This bio-assisted remediation technique is increasingly applied in global ecological engineering. Agricultural systems also benefit from mycorrhizal networks. Traditional tillage practices often disrupt natural fungal networks, leading to diminished soil fertility. Sustainable agriculture prioritizes preserving these networks by reducing tillage and implementing crop rotation systems to maintain healthy mycorrhizal symbiosis. This approach reduces chemical fertilizer use and enhances crop resilience. Climate change poses new challenges to mycorrhizal networks. Rising temperatures and shifting CO2 concentrations impact the symbiotic relationship between fungi and plants. Some fungal species may decline as a result, while others may gain an advantage. These dynamic shifts will reshape future ecosystems. Studying mycorrhizal networks requires innovative approaches. Scientists use stable isotope labeling to track nutrient flow within the network. Molecular biology techniques help identify the species and functions of different fungi within the network. These studies continually reveal the network's complexity. Mycorrhizal networks also inspire human technology. Their distributed, decentralized operational model offers new approaches for network communication and logistics management. Nature always provides us with the wisest insights, Protecting mycorrhizal networks safeguards the future of forests. When we stroll through forests admiring towering trees, remember their success depends on underground partners. This invisible network of connections is nature's most moving collaborative poem, reminding us of the profound truth that all things are interconnected. In today's pursuit of sustainable development, the wisdom of mycorrhizal networks is especially precious. It teaches us that cooperation triumphs over competition, and sharing leads to mutual benefit. This may be nature's most profound life lesson for us. Soil is not merely broken rock and decaying leaves. It is a complex ecosystem teeming with life. Fungi stand as one of the most vital engineers within this system. They construct and maintain soil in uniquely profound ways, transforming barren earth into fertile, teeming landscapes. Fungal involvement in soil formation begins on rock surfaces. Lichens, symbiotic unions of fungi and algae, serve as pioneering trailblazers. The organic acids they secrete gradually dissolve rock minerals, laying the groundwork for soil through this biological weathering process. Each year, lichens strip away a thin layer of minerals from the rock. Over thousands of years, this accumulation forms the initial soil layer. As the soil layer gradually builds, more types of fungi begin to colonize. Their mycelia form a natural network, weaving and growing between soil particles. These delicate filamentous structures possess remarkable mechanical strength, binding loose soil particles together to form stable aggregate structures. Soil aggregates are the fundamental building blocks of healthy soil. They act as the building blocks of soil, determining its physical properties. Fungal hyphae physically entwine and chemically bind tiny clay and sand particles into stable aggregates. This structure allows soil to maintain aeration, while retaining excellent water-holding capacity. Glycoproteins and polysaccharides secreted by hyphae form natural glue between soil particles. These adhesive substances help maintain aggregate stability, preventing soil structure breakdown, even under rainwater runoff or external disturbance. This self-repairing capacity is remarkable. In forest soils, fungal networks are exceptionally dense. With hundreds of meters of hyphae per gram of soil, these intricate networks act as natural reinforcing materials, significantly enhancing soil resistance to erosion. During heavy rains, soils with fungal networks effectively resist water and wind erosion, while protecting precious topsoil resources. Fungi also decompose organic matter to enrich soil with vital organic compounds. These compounds act as the soil's blood, not only supplying plant nutrients, but also improving soil physical properties by increasing buffering capacity and nutrient retention. During decomposition, fungi produce humus. This dark colloidal substance is critically important. It absorbs water and nutrient ions, preventing nutrient leaching, while darkening soil color, enhancing heat absorption capacity and promoting plant growth. Fungal activity also influences soil chemistry. By secreting organic acids, they alter soil pH, promoting dissolution of certain minerals, releasing nutrients required by plants. This biogeochemical process continuously regulates the soil's chemical environment. Different fungal types collaborate in specialized roles within the soil. Some specialize in breaking down cellulose. Others excel at decomposing lignin. Still others can degrade complex proteins. This functional diversity ensures organic matter is fully decomposed and recycled. Mycorrhizal fungi form vast networks throughout the soil. These networks connect not only plants, but also distinct soil microhabitats, functioning like an underground highway system, transporting nutrients, water, and signaling molecules. This networked operation enhances the efficiency of the entire soil system. Fungi also significantly shape soil pore structure. As mycelia grow, they form micropores within the soil. These channels facilitate air and water movement, while providing habitat for other soil organisms. Such biopores, compared to purely physical pores, are more conducive to the health of soil ecosystems. In arid regions, fungi play an especially critical role. Their mycelial networks help soil retain moisture by creating localized, humid microenvironments that provide valuable opportunities for plant seed germination and seedling growth. This moisture-retaining capacity is vital for ecological restoration. Fungi also help soil resist compaction. In modern agriculture, mechanical operations often cause soil compaction, impeding crop root growth and water infiltration. Soils rich in fungal networks effectively mitigate compaction issues due to their favorable aggregate structure and mycelium support. Soil fungi also participate in vital biogeochemical cycles. They play a dual role in the carbon cycle, releasing carbon dioxide through decomposition, while simultaneously fixing carbon into soil organic matter. This balance holds significant implications for global carbon equilibrium. In nitrogen cycling, certain fungi cooperate with nitrogen-fixing bacteria to create microenvironments conducive to nitrogen fixation. Other fungi directly decompose nitrogen-containing organic matter, converting organic nitrogen into inorganic nitrogen available for plant uptake. These processes are vital for maintaining soil fertility. Fungal fixation of heavy metals in soil also warrants attention. Certain fungi adsorb heavy metals via their cell walls, or form insoluble compounds, thereby reducing the bioavailability of heavy metals. This capability holds potential applications in remediating contaminated soils. In extreme environments like mine wastelands, fungi often pioneer ecological restoration. They tolerate harsh soil conditions, gradually improving soil physicochemical properties to create conditions for subsequent plant colonization. Modern agriculture has begun to recognize the role of fungi by applying fungal fertilizers or inoculating beneficial fungi. Soil quality improves and chemical fertilizer use decreases. These eco-friendly agronomic practices are being promoted globally. Protecting soil fungal diversity is crucial. Overcultivation and improper use of chemical pesticides and fertilizers can damage soil fungal communities, while adopting conservation tillage and cover crops promotes the healthy growth of soil fungi. When we hold a handful of healthy soil, the distinctive earthy fragrance we detect primarily stems from the metabolic products of actinomycetes and fungi. This aroma symbolizes the vitality of soil life. Against the backdrop of climate change, the role of soil fungi becomes even more prominent. They assist in storing greater amounts of carbon in the soil, enhancing the climate resilience of ecosystems. Understanding and protecting these unseen underground workers holds significant importance for addressing global environmental challenges. Through millions of years of evolutionary wisdom, fungi teach us how to coexist harmoniously with nature. They require neither heavy machinery nor chemical agents, but rely solely on exquisite biological design to construct and maintain fertile soil. This natural wisdom deserves our deep reflection and emulation. Each time we tread upon soft earth, we should express gratitude to these minute beings. They silently weave the underground web of life, sustenance for all life above ground. from the tiniest wildflowers to the tallest trees. All benefit from fungi's diligent work within the soil. Protecting soil means safeguarding these unseen heroes, preserving the home they depend on. This is not only an ecological imperative, but an investment in humanity's future. For healthy soil signifies healthy ecosystems and a more sustainable tomorrow. Fungi never exist alone in nature. They form intricate relationships with animals and other microorganisms. These connections transcend simple competition or predation, revealing the remarkable collaborative wisdom of the living world. From tiny insects to large mammals, from bacteria to other fungi, their interactions weave the rich tapestry of ecosystems. The partnership between leafcutter ants and fungi is a classic example. These industrious insects do not consume leaves directly. Instead, they cut leaves into pieces and carry them back to their nests, to serve as a growth medium for specialized fungi. Within their nests, the ants meticulously cultivate these fungi, clearing away unwanted microorganisms and regulating temperature and humidity. Under the ants' care, the fungi develop into nutrient-rich fungal balls, becoming the sole food source for the ant colony. This agricultural behavior has persisted for 50 million years. Termites and fungi share an equally remarkable collaboration. In tropical regions of Africa and Asia, termites construct magnificent mounds where they cultivate fungi. They gather plant material to build intricate fungal gardens. The fungi aid in breaking down plant fibers, enabling termites to digest lignin, while providing the fungi with an ideal growth environment. This symbiosis makes termites vital decomposers. Mammals also participate in fungal dispersal. Squirrels deliberately collect mushrooms and hang them on branches to dry. This storage behavior unexpectedly aids spore dispersal. North American grizzly bears consume large quantities of mushrooms in autumn. While storing fat for hibernation, they also transport spores to new territories. These large animals become long-distance dispersers for fungi. Fungal relationships with insects extend beyond mutualism. Some fungi parasitize insects, such as the renowned cordyceps. Spores attach to an insect's surface, germinate, and invade its body. Mycelium gradually fills the insect's body, eventually sprouting fruiting bodies from its head. This seemingly brutal process maintains ecological balance by controlling insect populations and preventing overpopulation. The fungus Trichoderma rhizae demonstrates fungi's sophisticated control abilities. After infecting fruit flies, it manipulates hosts to climb high and cling tightly to surfaces. After the host dies, spore capsules emerge from its body and spray spores in all directions. This precise behavioral manipulation ensures widespread spore dispersal. Within the decomposer alliance, fungi and bacteria collaborate seamlessly. Fungi break down complex substances like lignin, creating favorable conditions for bacterial activity. Bacteria then decompose the pre-processed material. Together, they achieve complete organic recycling. This division of labor enhances decomposition efficiency. Lichens represent the perfect fusion of fungi and algae. Fungi provide protective structures and support. Algae or cyanobacteria perform photosynthesis. This symbiotic entity thrives in extreme environments, from scorching deserts to frigid polar regions, demonstrating remarkable environmental adaptability. Lichens even serve as environmental quality indicator organisms. The chemical dialogue between fungi and bacteria is equally fascinating. Certain bacteria produce signaling molecules that stimulate fungi to generate antibiotics, while fungal metabolites influence bacterial communities. This allelopathic interaction regulates microbial balance, maintaining ecosystem health and stability. Within the human body, fungi also contribute to the microbiome. They collaborate with bacteria to maintain gut health. For instance, Saccharomyces cerevisiae helps regulate microbial balance. This endosymbiotic relationship is vital for host health, revealing another dimension of fungal-animal interactions. Fungi exhibit both cooperation and competition with other fungi. Some species form mycelial networks to share resources and information, while others wage chemical warfare for territory, producing antibiotics to inhibit competitors' growth. This dynamic equilibrium sustains ecosystem vitality. Fungal relationships with plants extend beyond mycorrhizae. Some endophytic fungi reside within plant tissues, assisting hosts in resisting pests and diseases, enhancing drought tolerance. These unseen allies quietly bolster plants' competitive edge for survival. Within aquatic ecosystems, fungi establish unique partnerships with aquatic animals. Certain freshwater sponges harbor symbiotic fungi, assisting in processing organic particles in water. This collaboration purifies aquatic environments. Fungi even develop special relationships with birds. North American jays feed on pine nuts, yet consistently bury more seeds than they need. These forgotten seeds germinate with fungal assistance, while the fungi obtain nutrients from the seedling's roots, forming a remarkable tripartite symbiosis. Microbial collaborations often remain unseen. The alliance between nitrogen-fixing bacteria and fungi is a prime example. Fungi provide living space for bacteria, while bacteria supply nitrogen sources for fungi. This cooperation is especially vital in nitrogen-poor environments. The chemical communication between fungi and animals is fascinating. Certain mushrooms produce pheromone-like substances to attract specific insects that aid in spore dispersal. This sophisticated chemical signaling system is the result of long-term coevolution in the field of biological pest control.
Humans la baraja fungal-insect relationships.
To develop eco-friendly pest management methods, such as using Boveria bachiana to combat agricultural pests, reducing reliance on chemical pesticides. Fungal partnerships with microorganisms also drive advancements in biotechnology. By studying these interactions, scientists develop novel fermentation processes and waste treatment technologies. These intricate biological relationships reveal that no organism exists in isolation in nature. Every life form is interconnected through countless links with others. Fungi are both participants and shapers in these relationships. Understanding the significance of these partnerships not only satisfies scientific curiosity, but also offers insights for protecting ecosystems. When we disrupt a particular fungus, we may impact the entire network of relationships, with unforeseen consequences. Under climate change pressures, these biological partnerships become even more precious. They enhance ecosystem resilience, helping communities adapt to environmental shifts. Protecting biodiversity means safeguarding these intricate networks. The history of fungal interactions with animal microbes is an epic of coevolution. Since the dawn of terrestrial ecosystems, they have jointly shaped our world. This ancient and enduring collaboration deserves our respect and protection. As we wander through nature, these subtle partnerships may remain unseen, yet they persistently operate, sustaining our planet's vitality and equilibrium. This reminds us to cherish every life form, for even the most inconspicuous organisms may fulfill irreplaceable ecological roles. The evolutionary history of fungi spans a billion-year epoch. They were among the earliest life forms to leave the oceans and conquer the land. Molecular clock studies indicate that the common ancestor of fungi and animals lived in the Proterozoic oceans approximately 1 billion years ago. This ancestor was a single-celled flagellate organism, capable of free swimming in aquatic environments. The earliest direct fossil evidence of fungi comes from Canada, in strata dating back approximately 1 billion years, where scientists discovered traces of filamentous microorganisms. These minute thread-like organisms may represent fungal ancestors, living within ancient marine sediments and utilizing chemical processes to obtain energy. By the Cambrian explosion, fungi had already begun exhibiting diversity. Fossils with mycelial structures were found in strata dating to 530 million years ago. Fossils with hyphal structures were discovered. These early fungi inhabited shallow marine environments, forming simple symbiotic relationships with algae and bacteria. When plants began colonizing land, fungi were already prepared. During the Ordovician period, approximately 470 million years ago, fungi established connections with the root systems of primitive plants. This ancient mycorrhizal symbiosis helped plants adapt to the arid terrestrial environment and ushered in a new era for terrestrial ecosystems. Fossils from the Silurian period reveal fungi had developed intricate mycelium systems, capable of decomposing organic matter on rock surfaces.
Accelerating soit la formation process.
These pioneer fungi created suitable growth environments for subsequent plant communities. The Devonian era is known as the Golden Age of fungi, featuring giant fungi like the genus Pterophorus, reaching heights of 8 meters, forming distinctive landscapes in prehistoric forests. These giant fungi lack true root systems, instead absorbing nutrients through subterranean mycelium networks. Fungi faced new challenges during the Carboniferous period, as vast forests covered the earth, requiring massive plant biomass decomposition. Fungi evolved novel enzyme systems, particularly lignin-degrading capabilities, establishing them as pivotal regulators of the carbon cycle. The Permian mass extinction event witnessed a brief fungal resurgence, as environmental upheaval wiped out most life forms. Fungi rapidly seized dominance. Fossil records reveal a dramatic increase in fungal spore counts within strata during this period. Early Mesozoic fungi began diversifying, giving rise to ancestors of modern major groups. Basidiomycota and Ascomycota diverged during this era, each developing distinct reproductive strategies. These evolutionary innovations laid a solid foundation for fungal diversity. The rise of flowering plants in the Cretaceous presented new opportunities for fungi. Many fungi established obligate symbiosis with angiosperms. This coevolutionary relationship persists today, shaping the landscape of modern terrestrial ecosystems. Cenozoic fungi continue evolutionary innovation. With the proliferation of mammals and birds, some fungi develop strategies reliant on animal dispersal, producing fruiting bodies with attractive odors and colors. This evolutionary direction enhanced mutualistic relationships between organisms. Molecular systematics research has revealed many mysteries on the fungal evolutionary tree. Certain groups previously classified as fungi, such as oomycetes, are actually more closely related to algae. Meanwhile, microsporidia have been confirmed as specialized fungi. Fungal genome studies indicate their closer kinship to animals than to plants, with significant genetic similarities, particularly in cellular signaling and metabolic pathways. These discoveries reshape our understanding of the tree of life. Fungi's evolutionary journey brims with ingenuity. Over billions of years, they developed unique survival strategies. From deep seas to mountain peaks, from equators to polar regions, ubiquitous fungi bear witness to Earth's transformations. They also participate in shaping the planet's destiny. At nearly every pivotal turning point in Earth's history, fungi have contributed and played a role. They may not be the protagonists on stage, but they remain vital driving forces behind the scenes. Through collective action, these microscopic organisms altered the planet's chemical environment and ecological patterns. During the Great Oxygenation Event, fungi played a special role. Though still in their evolutionary infancy, they could already influence the chemical equilibrium of their surroundings through metabolic processes, contributing to the eventual stabilization of atmospheric oxygen levels. During the Cambrian explosion, fungi began to emerge as decomposers. They broke down the remains of dead organisms, facilitating the recycling of nutrients and sustaining the functioning of early ecosystems. When plants began colonizing land during the Ordovician period, fungi played a crucial supporting role. Through mycorrhizal symbiosis, they helped plants overcome challenges like drought and nutrient uptake. This collaboration accelerated the establishment of terrestrial ecosystems. The Devonian period witnessed a particularly spectacular fungal event. Giant fungi like Pteropogon covered the land. These organisms, reaching several meters in height, formed the earliest forest landscapes. Through their powerful decomposition capabilities, they continuously altered soil chemistry. During the coal formation process in the Carboniferous period, fungi played a dramatic role. At this time, lignin-degrading enzymes were not yet fully developed, leaving vast plant remains incompletely decomposed, ultimately forming coal, today's vital human energy source. During the Permian mass extinction, fungi demonstrated remarkable adaptability. While most animals and plants suffered mass die-offs,
Fungi experiencent la population explosion.
decomposing widespread organic debris, sustaining the fundamental functions of ecosystems. In the early Triassic, fungi aided ecosystem recovery by stabilizing soil through their mycelial networks and creating conditions for the reestablishment of plant communities. Fossil records from this period indicate a rapid recovery and surpassing of previous levels of fungal diversity. During the Cretaceous flowering plant revolution, fungi participated in a process of coevolution. Many orchids became entirely dependent on specific fungi for nutrition. This obligate symbiosis persists to this day, demonstrating exquisite coordination throughout evolution. During the Cretaceous-Paleogene extinction event, fungi again proved their resilience. Amidst the environmental catastrophe triggered by asteroid impact, they recovered faster than many animals and plants, emerging as pioneers in ecological reconstruction. In the early Cenozoic, fungi continued expanding their influence. With the rise of mammals, some fungi developed novel dispersal strategies, utilizing animal digestive systems for propagation. This mutualistic relationship persists to this day. During the Quaternary glacial periods, fungi faced severe challenges. Glacial advances and retreats altered their distribution patterns. Yet fungi successfully weathered multiple glacial tests through spore dispersal and dormancy strategies, and rapidly revived during interglacial periods. Since the Industrial Revolution, human activities have introduced novel factors influencing fungal evolution. Certain fungi rapidly adapted to anthropogenic environments. finding new ecological niches in urban areas and farmlands, demonstrating remarkable evolutionary plasticity. Fungi also regulate geochemical cycles. Through decomposition, they influence carbon cycling, and via symbiotic relationships, they participate in nitrogen cycling. These seemingly minor individual actions accumulate into global-scale ecological impacts. Fungi play an indispensable role in soil formation. From rock weathering to humus formation, every step bears fungal contributions. Over hundreds of millions of years, they transform barren rock into fertile soil. Fungi face new challenges amid climate change. Rising temperatures and shifting CO2 levels are altering fungal distribution and activity. Some species may decline, while others gain an advantage. This dynamic shift will impact entire ecosystems. Looking back at Earth's history, we clearly see that fungi, though microscopic, wield profound influence. They have helped shape today's natural world and will continue to impact the planet's future. Understanding these pivotal historical events aids us in better protecting our planet. As Earth's most vital decomposers, fungi play a crucial role in the global carbon cycle. They act as nature's carbon cycle engineers, silently regulating atmospheric carbon balance, influencing global climate trends. Each year, plants on Earth absorb vast amounts of carbon dioxide through photosynthesis, converting it into organic matter. When these plants die, the carbon stored in their remains must return to the atmosphere. This process relies primarily on fungi. By decomposing dead organic matter, fungi release sequestered carbon as carbon dioxide, completing the final link in the carbon cycle. Different fungal species exhibit distinct roles in carbon cycling. White rot fungi specialize in decomposing lignin, while brown rot fungi primarily break down cellulose. Other fungi decompose simpler organic compounds. This functional differentiation ensures decomposition efficiency and maintains the stable operation of the carbon cycle. During decomposition, fungi face carbon allocation choices. A portion of carbon is used for their own growth and metabolism, converted into fungal biomass for temporary storage. Another portion is released into the atmosphere as carbon dioxide through respiration, while some carbon is converted into stable compounds, stored long-term within soil organic matter. Soil serves as the primary arena where fungi regulate the carbon cycle. In forest and grassland soils, fungal mycelium networks store vast amounts of carbon. This carbon exists within the cell walls of mycelium. Chitin, a stable carbon-containing compound, can persist in soil for extended periods. Mycorrhizal fungi play a unique role in carbon storage. They obtain carbohydrates from plants, while simultaneously storing carbon within their extensive mycelial networks. Research indicates that mycorrhizal fungal biomass may constitute 1/3 of total soil microbial biomass. These subterranean carbon reservoirs are astonishing in scale. Fungi in different ecosystems contribute to carbon cycling in varying ways. In northern forests, fungal-dominated decomposition proceeds slowly, resulting in substantial carbon storage within litter layers. Conversely, decomposition proceeds rapidly in tropical rainforests, enabling faster carbon turnover within ecosystems. Climate change is influencing fungal decomposition activities. Temperature increases typically accelerate decomposition processes, releasing greater amounts of carbon into the atmosphere. This positive feedback effect may exacerbate global warming. However, the reality is more complex, as rising temperatures also alter fungal community structures. Increased atmospheric carbon dioxide concentrations also affect fungal function. Some fungi grow faster in carbon-rich environments, enhancing their carbon sequestration capacity, while others may alter metabolic pathways, influencing carbon release rates. Extreme climate events like drought significantly alter fungi's carbon regulation role. Under drought conditions, fungal activity declines, decomposition slows, and carbon release decreases. Simultaneously, plant growth is inhibited, disrupting the entire ecosystem's carbon balance. Fungal regulation of the carbon cycle extends beyond decomposition. They also indirectly influence carbon sequestration and release through interactions with other organisms. For instance, mycorrhizal fungi enhance plant photosynthesis, promoting greater carbon dioxide uptake. These indirect effects are equally significant. Within marine ecosystems, fungi participate in carbon cycling processes by decomposing organic particles, influencing the dynamics of marine carbon pools. Although research remains relatively scarce, the role of marine fungi cannot be overlooked. Human activities are altering fungi's carbon regulation functions. Deforestation reduces fungal habitats. Agricultural practices transform soil fungal communities. Pollutant emissions impact fungal metabolic activity. These disturbances may disrupt natural carbon equilibrium. Understanding fungi's role in the carbon cycle is crucial for predicting climate change. Scientists use techniques like isotope labeling to track carbon flow within fungal networks. These studies help build more accurate climate models. Protecting fungal diversity holds climate significance. Rich fungal communities enhance ecosystem resilience and help mitigate impacts of climate change. For instance, certain fungi boost plant drought tolerance, indirectly enhancing carbon sequestration. In carbon neutrality strategies, fungi offer natural solutions. By restoring healthy fungal communities, soil carbon storage capacity is enhanced. This nature-based solution is cost-effective and delivers multiple ecological benefits. Certain fungi possess unique carbon fixation abilities. For instance, fungi in some lichens directly utilize atmospheric carbon dioxide, though far less efficient than plants. This diversity showcases nature's ingenuity. Fungal carbon regulation functions also face threats. Climate change may cause certain fungal species to disappear, disrupting the original carbon cycle balance. Protecting fungal habitats means safeguarding Earth's climate regulation mechanisms. Fungi also play a role in urban environments. They decompose organic matter produced by urban greenery, influencing the carbon balance of urban ecosystems. Rational utilization of fungal functions can enhance cities' sustainable development capabilities. Looking ahead, fungi may play an even greater role in climate governance by artificially cultivating specific fungal species to enhance soil carbon sequestration, or utilizing fungi to process organic waste to reduce greenhouse gas emissions. Through billions of years of evolutionary wisdom, fungi teach us a simple yet profound truth. Every member of nature matters. Though seemingly insignificant, fungi influence climate on a global scale. This connection between the macro and micro deserves our deep reflection and respect. When we focus on climate change, we should not only see factories and cars, but also the fungi silently working in the soil beneath our feet. They are both witnesses to climate change and vital forces in addressing its challenges. Fungi possess a unique feeding strategy in nature. They neither ingest food like animals, nor perform photosynthesis like plants. Instead, They employ a unique strategy of external digestion. At the core of this strategy lies the use of enzymes. Enzymes are biological catalysts produced by fungi. They function like precision molecular tools, capable of breaking down complex organic matter into simpler molecules. Each enzyme has a specific target, fitting together with its substrate as precisely as a key in a lock. This specificity ensures the orderly progression of the decomposition process. Fungi secrete a diverse array of enzymes. Cellulase specifically breaks down cellulose. Ligninase tackles stubborn lignin. Protease handles protein decomposition. These enzymes collaborate like a precision-engineered decomposition team, gradually dismantling complex organic matter. The secretion of enzymes is a highly refined process. Fungi release enzyme molecules through the tips of their hyphae. These enzymes then diffuse into the surrounding environment. Upon binding to substrates, they initiate catalytic reactions. This entire process enables fungi to acquire nutrients from distant locations. Without needing to relocate, lignin degradation presents the most formidable challenge. This complex polymer constitutes plant cell walls and exhibits exceptional resistance to decomposition. Peroxidases produced by white rot fungi can cleave lignin's aromatic ring structures, representing one of nature's most remarkable decompositional techniques. When breaking down cellulose, fungi employ different strategies. Fungi secrete endocellulases that randomly cleave cellulose chains. Exocellulases sequentially release glucose units from the chain ends. Beta-glucosidases then fully degrade these small fragments. This multi-enzyme system ensures complete utilization of cellulose. Proteases perform equally intricate work by cleaving peptide bonds in proteins to break down complex proteins into amino acids. These amino acids can be directly absorbed and utilized by fungi as building blocks for their own cells. The greatest advantage of external digestion lies in fungi's ability to process food sources vastly larger than themselves. A single mycelium cluster can decompose an entire fallen leaf, or even penetrate the interior of massive dead logs, gradually breaking down the entire structure from the inside out. This process is highly sensitive to environmental conditions. Temperature affects enzyme activity.
pH determine enzyme efficiency.
Humidity influences enzyme diffusion rate. Fungi perceive these environmental factors and adjust enzyme secretion levels and types accordingly. Different fungal species possess specialized enzyme systems. Some excel at breaking down plant matter. Others specialize in degrading keratin. Still others can decompose synthetic materials like plastics. This diversity allows fungi to occupy diverse ecological niches. During decomposition, fungi demonstrate remarkable intelligence. When encountering easily degradable substances, they secrete basic enzymes to rapidly acquire nutrients. When faced with complex materials, they activate specialized enzyme systems. This regulatory ability conserves energy expenditure. Enzymes serve purposes beyond nutrient acquisition. Certain enzymes function defensively. Fungi secrete antibiotic enzymes to inhibit competitors' growth, protecting their food sources and territories. Humans have long harnessed fungal enzyme systems. In traditional brewing and bread making, yeast enzymes convert sugars into alcohol and carbon dioxide, an application dating back millennia. Modern industrial applications of fungal enzymes are even more extensive. Cellulase is used in textile processing, protease in detergent manufacturing, amylase in the food industry. These biocatalysts are more environmentally friendly than chemical methods. Fungal enzymes play a vital role in the paper industry. Xylanase aids in bleaching pulp, reducing the use of chemical bleaches, lowering costs while protecting the environment. The pharmaceutical field also benefits from fungal enzymes. Glucose oxidase is used for blood glucose testing. Chymotrypsin is used to dissolve blood clots. These enzyme preparations have saved countless lives. Fungal enzymes demonstrate remarkable capabilities in environmental protection. They can degrade pesticide residues, break down petroleum pollutants, purify industrial wastewater, providing green solutions for ecological restoration. Recent research brings even greater excitement. Scientists have discovered that certain fungal enzymes can break down plastic products, offering hope for tackling white pollution. Enzyme production itself has become an industry. Fungi are mass-cultivated through fermentation engineering, and their enzyme preparations are extracted and purified. This process itself harnesses the unique properties of fungi. Understanding the external digestion mechanism of fungi not only satisfies scientific curiosity, but also provides insights for sustainable development. These microscopic organisms, with their chemical ingenuity, demonstrate nature's exquisite design. Each time fungi decompose dead wood, it becomes a vivid chemistry lesson. They accomplish the most complex transformations through the gentlest means. This wisdom deserves deep study by humanity and application in technological innovation. Alexander Fleming's serendipitous discovery in 1928 unleashed a new chapter in modern medicine. When he observed penicillium mold inhibiting Staphylococcus growth, he likely never imagined this finding would transform the world. The birth of penicillin heralded the dawn of the antibiotic era. Penicillin belongs to the beta-lactam class of antibiotics. It disrupts bacterial cell wall synthesis, causing bacteria to collapse and die from structural failure. This specific mechanism renders it harmless to mammalian cells, making it an ideal antibacterial agent. With the large-scale production of penicillin, countless infectious diseases were effectively controlled. Once fatal illnesses like pneumonia, puerperal fever, and sepsis became curable common ailments. Human life expectancy consequently increased significantly. Penicillin's success inspired scientists to seek more antibiotics. Waxman discovered streptomycin from Streptomyces bacteria. This marked the first effective treatment for tuberculosis, ushering in a new era of anti-tuberculosis therapy, and led to the coining of the term antibiotic. Beyond bacterial infections, fungi also yield antifungal agents. Amphotericin B, derived from Streptomyces nodosus, effectively treats deep fungal infections, though it carries some nephrotoxicity, remaining the first-line treatment for certain lethal fungal infections to this day. Cyclosporin, discovered among fungal metabolites, revolutionized the field of organ transplantation. This compound from Trichoderma polyphos selectively suppresses T-cell activity to prevent transplant rejection, transitioning organ transplantation from experimental to routine treatment. Tacrolimus represents another significant discovery. This compound from Streptomyces succubensis exhibits 100-fold greater potency than cyclosporin, offering more options for transplant patients and improving the quality of life for countless patients. The discovery of levastatin is equally commendable. This metabolite from Aspergillus terreus effectively inhibits cholesterol synthesis and became a vital drug for treating hypercholesterolemia and preventing cardiovascular and cerebrovascular diseases. Fungi have also made significant contributions to anti-cancer drug development. Taxol, derived from endophytic fungi, has become an effective treatment for ovarian and breast cancers. Its unique mechanism of action inhibits tumor cell division by stabilizing microtubules. The discovery of penicillin offers profound insights. Fleming originally studied influenza viruses, when a chance bacterial contamination altered his research direction. This reminds us that scientific breakthroughs sometimes require luck. But more crucially, a mind adept at observation and critical thinking. As antibiotic misuse becomes increasingly severe, fungi once again offer potential solutions. Scientists seek novel antibiotics from fungi to address the escalating challenge of drug resistance. The diversity of fungal secondary metabolites provides invaluable resources for drug development. Statistics indicate that approximately 50% of clinically used drugs directly or indirectly originate from fungi and other microorganisms. This figure continues to grow. Modern drug discovery technologies accelerate the process. Genomics aids in identifying gene clusters within fungi to predict potentially active compounds. Synthetic biology enables large-scale production of these compounds. Fungally derived drugs not only treat diseases, but also serve diagnostic and preventive purposes. Glucose oxidase is used in blood glucose test strips, while fungal polysaccharides function as immunomodulators. These applications expand fungi's medicinal value. Fungi have long been widely used in traditional medicine. Medicinal fungi like Ganoderma lucidum, Porreococcus, and Cordyceps sinensis have been utilized for millennia in Eastern medicine. Modern research is validating their pharmacological effects and providing scientific evidence for traditional knowledge. Fungal drug development faces challenges. Many fungi fail to express their full metabolic potential under laboratory conditions. Activating these silent gene clusters has become a current research focus. As understanding of fungi deepens, more medicinal value is being discovered. Certain fungal metabolites exhibit antiviral and anti-Alzheimer's disease activities, offering new directions for future drug development. Fungi not only provide drugs themselves, their metabolic pathways also inspire chemical synthesis. Scientists mimic fungal biosynthetic pathways to develop more environmentally friendly and efficient production processes. In the era of personalized medicine, fungal drugs demonstrate new potential. By genetically engineering fungi to produce drugs tailored to specific patients' needs, this precision medicine approach holds vast promise. Looking back at the history of antibiotics and pharmaceutical development, fungi have made an indelible contribution. From the accidental discovery of penicillin to modern targeted drug development, fungi continue to support human health. Looking ahead, as drug resistance intensifies and climate change alters disease distribution, the importance of fungal medicinal resources becomes increasingly evident. Protecting fungal diversity is safeguarding the future of human health. Fungi have deep roots in human culinary culture. The earliest fermentation practices date back to the Neolithic era, when ancient people accidentally discovered moldy grains could produce unique flavors and pleasurable effects. The food partnership between humans and fungi began. Brewing is one of the oldest fungal applications. Yeast converts sugars into ethanol and carbon dioxide. This seemingly simple biochemical process has fostered a rich and diverse wine culture. From beer and wine to spirits, fungi participate in brewing the world's major alcoholic beverages. Bread making similarly relies on the power of fungi. Yeast produces carbon dioxide within dough. to form a soft, porous bread structure, while generating flavor compounds like alcohols and esters that impart bread's distinctive aroma and texture. Soy sauce production showcases the subtlety of fungal fermentation. Aspergillus oryzae breaks down proteins in soybeans and wheat into amino acids and peptides, while converting starch into sugars. After months or even years of fermentation and maturation, it ultimately forms a savory, rich condiment. Fungi are indispensable in cheese making. Penicillium molds participate in blue cheese ripening, imparting its distinctive flavor and texture. Penicillium albigenes aids the maturation of soft cheeses. These fungal fermented cheese products have become integral components of Western culinary culture. Eastern cuisines feature a rich variety of fermented fungal foods. Miso, made by fermenting soybeans with Aspergillus oryzae, serves as a fundamental seasoning in Japanese cuisine. Red yeast rice, produced by fermenting rice with Menascus purpureus, functions both as a natural pigment and a health food. In modern food industries, fungi find even broader applications. Fungally produced citric acid serves as an acidity regulator in beverages and foods, with annual production exceeding 2 million tons, demonstrating the industrial application of fungi. Fungal enzyme preparations are ubiquitous in food processing. Amylase is used in syrup production. Protease is used for meat tenderization. Pectinase aids in juice clarification. These enzyme preparations enhance production efficiency and improve product quality. Fungal polysaccharides, such as shiitake polysaccharides and Ganoderma polysaccharides, serve as natural food additives, providing functions like thickening, stabilizing, and emulsifying, while also offering health benefits. Single-cell protein represents another significant contribution from fungi. Utilizing fungal mycelia like fusarium, produces high-protein food ingredients. This microbial protein production is highly efficient, with a far smaller environmental impact than traditional livestock farming. Fungal applications are equally extensive in industrial sectors. Enzyme preparations produced by fungi are utilized in the textile industry. Cellulase is employed for denim biostone washing, replacing traditional pumice to reduce equipment damage. In the papermaking industry, xylanase aids bleaching, reducing the use of chlorine-based bleaching agents, lowering costs while protecting the environment. Lactase is used for delignification of pulp, enhancing paper whiteness and quality. Biological detergents contain fungal proteases and lipases, effectively remove stains at low temperatures, conserve energy consumption. These enzyme preparations are biodegradable, more environmentally friendly than chemical detergents. Fungi play an increasingly vital role in biofuel production. Cellulase converts agricultural waste into fermentable sugars. Yeast fermentation produces bioethanol. This second-generation biofuel does not compete with food crops, ensuring sustainability. Environmental remediation represents an emerging application for fungi. Certain fungi degrade petroleum pollutants for crude oil spill treatment. White rot fungi decompose recalcitrant organic pollutants, like polycyclic aromatic hydrocarbons. Fungi also play a role in mining industries. Fungal secretions leach metals from ores through this biometallurgy technique that proves more eco-friendly and energy efficient than conventional methods. Plastic degradation represents a frontier application for fungi. The recently discovered Aspergillus tabarensis can decompose polyurethane plastics, offering new hope for combating white pollution. Fungal extracts are highly valued in the cosmetics industry. Ganoderma lucidum and Cordyceps sinensis extracts serve as antioxidant ingredients in skincare products. Fungal fermentation products function as natural preservatives, replacing chemical preservatives. Traditional brewing industries are revitalized with modern technological support. Through selective breeding of superior strains and optimization of process parameters, product quality and stability are enhanced, while preserving traditional flavor characteristics. Fungi also play a role in food safety. Nattokinase produced by fungi serves as a natural preservative for food preservation, inhibiting mold and yeast growth. With advances in biotechnology, fungal applications in industry continue to expand. Genetically engineered fungi can now produce a broader range of valuable compounds, including pharmaceuticals, flavorings, and specialty chemicals. The sustainable nature of industrial fungal applications aligns with circular economy principles, utilizing agricultural and forestry waste as substrates for high-value product production, enables efficient resource utilization. From the dinner table to the factory floor, from tradition to modernity, fungi, with their unique biological properties, offer diverse solutions for human life. This ancient biological partner is demonstrating renewed vitality with technological support. Looking ahead, with the advancement of synthetic biology, fungi may become cellular factories, producing goods currently reliant on fossil-based raw materials, offering biomanufacturing solutions for sustainable development. The fungal world is a treasure trove of colors, from deep blacks to vibrant reds, from soft yellows to mysterious blues. These hues all originate from natural pigments produced by fungi. These pigments are not only beautiful but also possess significant biological functions. Meniscus-derived meniscus pigment is the most renowned fungal pigment, with a millennia-long history of use in China. This vivid red pigment boasts exceptional stability, widely applied in meat products, soy products, and alcoholic beverages, offering greater safety and health benefits compared to synthetic dyes. The black pigment produced by Aspergillus niger is equally significant. This pigment protects fungal cells from ultraviolet radiation and oxidative stress. It serves as a natural colorant in human cosmetics, while also providing antioxidant properties. The yellow pigment secreted by Aspergillus flavus has diverse applications. It functions as a food colorant in dairy products and beverages, and as a natural dye in the textile industry. These pigments are biodegradable and more environmentally friendly than synthetic dyes. The blue pigment produced by Penicillium species is uniquely distinctive. This pigment is pH sensitive and exhibits different colors at varying acidity levels. It can be used to create acid-base indicators and plays a role in educational experiments and industrial testing. The biosynthesis pathways of fungal pigments are complex and intricate. Typically, multiple enzymes collaborate to complete the process. Gene clusters control the entire synthesis. This precise regulation ensures pigments are synthesized at the appropriate time and location. Pigments are vital for fungal survival. Some pigments aid in absorbing specific wavelengths of light and converting them into energy for metabolism. Others provide protection against competition from other microorganisms. Still others help attract insects to assist in spore dispersal. Fungal pigments demonstrate distinct advantages in the food industry. High safety profile validated by long-term consumption. Excellent stability with minimal processing interference. Some possess nutritional and health benefits, e.g. monocolin in red yeast rice pigment exhibits lipid-lowering effects. The textile industry is rediscovering fungal dyes. Compared to traditional chemical dyes, fungal dyes are renewable and biodegradable, with minimal pollution during production, aligning with green manufacturing principles. The cosmetics industry favors fungal pigments for their mild, low irritation properties and simultaneous skincare benefits. For instance, Aspergillus niger pigment absorbs ultraviolet rays and serves as a natural sunscreen ingredient. In artistic creation, fungal pigments reveal unique value. Some artists cultivate fungi specifically to obtain natural pigments of varying hues. These bioartistic creations awaken reverence for nature. The biotechnological potential of fungal pigments extends far beyond this. Scientists employ genetic engineering techniques to modify fungal pigment synthesis pathways, enhancing yields or creating novel colors. This directed evolution technology broadens the application scope of fungal pigments. Environmental monitoring represents another emerging field. Certain fungal pigments exhibit sensitivity to environmental pollutants, changing color with pollutant concentration levels. They can be developed into biosensors for water and air quality monitoring. In medicine, fungal pigments demonstrate therapeutic potential. Research reveals certain pigments possess anti-inflammatory, antioxidant, and even anti-tumor activity, providing novel lead compounds for drug development. Agricultural applications also hold vast promise. Fungal pigments can serve as biopesticides, repelling pests through color, or function as growth regulators, enhancing crop stress resistance. In industrial biotechnology, fungi serve as cell factories, capable of producing pigments themselves, while enzymes from their pigment synthesis pathways can also synthesize other compounds. Extraction techniques for fungal pigments continue advancing, from traditional solvent extraction to supercritical fluid extraction and bioprocessing methods. These innovations enhance efficiency while reducing environmental impact. With synthetic biology's development, fungal pigment production enters a new phase. Scientists transfer pigment synthesis genes into easily cultivable microorganisms to achieve efficient, large-scale production. Research into fungal pigment stability deepens through techniques like microencapsulation to enhance resistance to light and heat and broaden application scope. In cultural heritage preservation, fungal pigments play a role by binding heavy metal ions to form stable complexes for artifact conservation and treatment. The food packaging industry has begun adopting fungal pigments as indicators for smart packaging. When food freshness changes, the packaging color shifts accordingly to alert consumers. Fungal pigments in cosmetics continue to innovate through nanotechnology processing to enhance pigment dispersion and stability while boosting functionality. The application of fungal dyes in textiles is forming a complete industrial chain, from fungal cultivation to pigment extraction and fabric dyeing processes, with the entire workflow achieving green production. Biotechnology companies are actively developing novel applications for fungal pigments. Some firms focus on pharmaceutical uses, others concentrate on food additives, while additional players explore industrial dye markets. This diversification demonstrates the immense potential of fungal pigments. Fungal pigments offer unique contributions to environmental protection. Certain pigments adsorb heavy metal ions for wastewater treatment. Some degrade organic pollutants, improving soil quality. Academic research continually uncovers novel properties of fungal pigments. Recent discoveries reveal certain deep-sea fungal pigments possess exceptional fluorescent characteristics, with broad application prospects in bioimaging and detection. The biosafety of fungal pigments remains a key focus. Through toxicological studies and clinical trials, application safety is rigorously ensured. This meticulous approach fosters healthy industry development. Future applications of fungal pigments may expand across diverse fields, from energy to materials, from electronics to aerospace. The potential of these natural pigments is only beginning to be recognized, reflecting on humanity's history with pigments, from mineral pigments to plant dyes, to fungal pigments. We are advancing toward a more sustainable future. Fungi, with their unique biosynthetic capabilities, offer us a rich palette of natural solutions. Looking ahead with technological advancements, fungal pigments will continue to surprise us. The world of color created by these microscopic organisms not only beautifies our lives, but also charts new paths toward sustainability. In the dialogue between science and nature, fungal pigments are writing a remarkable chapter. At the dawn of human civilization, fungi had already entered our lives. Prehistoric humans, during their foraging activities, gradually came to recognize these wondrous organisms. The discovery of the European iceman Otzi proves that humans were using medicinal fungi 5,000 years ago. His leather pouch contained various mushrooms, likely for medicinal or religious purposes. Ancient Egyptians held fungi in deep reverence. They regarded mushrooms as gifts from the god of life. Mushroom-shaped motifs frequently appear in the tomb murals of pharaohs. The builders of the pyramids may have consumed mushrooms to gain extra nutrition and energy. In ancient Greek civilization, mushrooms held exalted status. Philosophers observed their miraculous growth, believing them born from Zeus' thunderbolts striking the earth. This perception reflected ancient peoples' mystical understanding of mushrooms' sudden emergence. During the Roman Empire, mushrooms became luxury items. Nobles particularly cherished edible fungi, even establishing dedicated cultivation gardens. The renowned gastronome Apicius detailed cooking methods for various mushrooms. In his culinary treatise, Central American civilizations held unique interpretations of fungi. The Aztecs referred to certain hallucinogenic mushrooms as the flesh of the gods, using them in religious ceremonies. They believed these sacred mushrooms enabled direct communication with the divine realm. In ancient China, fungi were documented early on. The Divine Farmer's Classic of Materia Medica documented the efficacy of Lingzhi mushrooms, classifying them as premium medicinal herbs. Subsequent medical texts provided detailed descriptions of medicinal fungi, forming a unique system of Chinese medicinal mycology. Shiitake cultivation has a long history in Japanese culture, with earliest written records dating back to the 1st century AD. People employed log cultivation methods to produce shiitake mushrooms. This traditional technique persists to this day, now recognized as a globally important agricultural heritage system. Medieval European fungal knowledge was rife with contradictions. Monks in monasteries meticulously studied and documented the morphological characteristics of various mushrooms, while folk legends associated mushrooms with witchcraft and magic. The Renaissance period witnessed breakthroughs in fungal research. Botanists began systematically classifying mushrooms and creating detailed botanical illustrations. These early scientific works laid the foundation for modern mycology. North American indigenous peoples possessed unique insights into fungi. They not only gathered mushrooms for food, but also utilized mycelium as tinder for fire. Certain tribes wove mushroom patterns into blankets. As family totems and cultural heritage, African tribes similarly employed fungi for centuries. They discovered that certain mushrooms could alter consciousness. Used in coming-of-age and healing ceremonies, this traditional knowledge passed down through generations formed a rich folk culture. In ancient Indian Ayurvedic medicine, various fungi were employed to treat ailments. Ancient texts documented mushrooms, energizing and immune-boosting properties. Mushrooms hold special status in Russian folklore. Mushroom foraging is a traditional autumn activity, inspiring numerous folk songs and tales. Mushrooms even appear on the painted patterns of traditional matryoshka dolls. In Norse mythology, mushrooms hold special significance. Legends tell of elves sheltering beneath them during rain, while fairy rings were believed to be traces of their dancing. These beautiful tales enrich folk culture. Renaissance artworks frequently featured mushroom imagery, from Bosch's fantastical paintings to Bruegel's rural scenes. Mushrooms appeared in diverse artistic forms. The 18th century witnessed flourishing mycological research. Linnaeus established a fungal classification system, though fungi were then classified within the plant kingdom. His work charted the course for subsequent studies. During the Industrial Revolution of the 19th century, fungal research achieved major breakthroughs. Pasteur discovered the microbial nature of fermentation. This discovery not only advanced science, but also transformed food industry production methods. Fungal research advanced rapidly in the early 20th century. The discovery of penicillin not only saved lives, but also changed humanity's understanding of microorganisms. During this period, mycology became an independent discipline. During wartime, fungal knowledge served a vital purpose. Soldiers learned to identify edible mushrooms, to address food challenges in the field. These practical skills saved countless lives. Mid-20th century research on psychedelic mushrooms sparked a cultural psychedelic revolution. Artists and musicians sought new creative inspiration. During this era, fungi gained unprecedented attention. In popular culture, modern science is rediscovering the value of traditional fungal knowledge. Ethnomycology, an emerging discipline, dedicates itself to documenting and studying indigenous peoples' traditional understanding and utilization of fungi across regions. Throughout history, fungi have consistently accompanied the development of human civilization. They serve as both food sources and medicines, cultural symbols and scientific subjects. This profound cultural connection continues to evolve and deepen today. Entering the 21st century, fungi have become even more intertwined with human life. These ancient biological partners are assuming new roles in modern society. Fungi are indispensable in the food industry. Billions consume daily yeast-leavened bread and steamed buns. This fundamental dietary practice relies on fungal fermentation. The rise of vegetarianism drives fungal protein development. Fungal protein produced from fusarium has become a key ingredient in meat alternatives. Its fibrous structure resembles muscle tissue, delivering a meat-like texture and mouthfeel. In the craft beer revolution, yeast is pivotal. Brewers select distinct yeast strains to create beers with varied flavor profiles. This precise control over microorganisms demonstrates the sophistication of modern food technology. The fashion industry embraces fungal materials. Mycelium leather emerges as a new favorite. This biomaterials production process is environmentally friendly, requiring no animal husbandry. and no toxic chemicals, aligning with sustainable development principles. Fungi revolutionize architectural design. Mycelium composites are used to manufacture biodegradable building fillers. These materials are lightweight yet strong, widely applied in temporary structures. The packaging industry is undergoing a fungal revolution. Packaging materials produced from mycelium are fully biodegradable, replacing plastics like polystyrene. Major corporations are adopting this eco-friendly packaging solution. Fungi excel in environmental remediation. Mushrooms purify contaminated soil, a process known as mycorrhizal remediation. White rot fungi break down pesticide residues, while other species absorb heavy metals, offering natural solutions for ecological restoration. Pharmaceutical R&D continues to rely on fungal resources. Beyond known antibiotics, scientists are exploring fungi for new anti-cancer drugs and immunomodulators. Fungal secondary metabolites remain a vital source of pharmaceuticals. The cosmetics industry values fungal ingredients. Extracts from Ganoderma lucidum and Tremella fusiformis have become key components in premium skincare products due to their moisturizing and antioxidant properties. These natural ingredients are highly favored by consumers. Fungal materials are emerging in home design. Furniture and planters crafted from mycelium are not only eco-friendly but possess unique aesthetic appeal. Designers create distinctive biodesign pieces. By controlling growth conditions, fungi play a role in waste management. They decompose organic waste, reducing landfill burdens. Certain mushrooms can even degrade hard-to-break-down materials like plastics. New applications emerge in mental health. Research indicates microdosing psychedelic mushrooms may aid in treating depression and PTSD. This novel therapy undergoes rigorous scrutiny. Fungi gain prominence in education. Schools introduce fungal science curricula to cultivate students' understanding of the microbial world. Outdoor mushroom identification activities become popular nature education formats. Mushroom cultivation thrives in urban agriculture. Urban residents cultivate oyster mushrooms on their balconies using coffee grounds and other waste. This circular economy model produces food while reducing waste. Tech startups focus on fungal applications, from biomaterials to environmental tech, from food innovation to pharmaceutical R&D. Fungus-related entrepreneurial projects attract increasing investment attention.
Fungi inspire artistic creation.
Bioartists create living artworks using mycelium that continuously grow and transform. This dynamic art form challenges traditional artistic concepts. The music industry also feels fungal influence. Some musicians claim consuming mushrooms helps them overcome creative blocks. Though controversial, it's undeniable. Fungi wield significant influence in popular culture. Mushroom identification becomes a pastime in outdoor pursuits. Hikers learn to recognize edible varieties, combining mushroom foraging with hiking excursions. Such activities foster deeper human-nature connections. Fungal technology holds significant importance in food safety. Nattokinase, produced by fungi, is widely used as a natural preservative, reducing the need for chemical preservatives. In the sustainability movement, fungi are regarded as vital allies. Their growth requires minimal resources, yet yields high-value products. This efficiency aligns with circular economy principles. Scientific research continues to uncover new fungal applications. From biobatteries to wastewater treatment, from air purification to building materials. The scope of fungal applications keeps expanding. In modern life, we may not realize how many times we rely on fungi daily. From breakfast bread to dinner soy sauce, from medications we take to products we use, fungi have become indispensable silent partners in our lives. Looking ahead as technology advances, the partnership between fungi and humans will deepen. This symbiotic relationship, spanning millions of years, is now writing a new chapter, facing global challenges of climate change and resource depletion. Fungi are demonstrating unique value as solutions. These ancient organisms, through their distinctive methods, offer fresh perspectives for human sustainable development. In waste management, fungi excel. They transform agricultural waste into resources. Fibrous materials like straw and corn cobs can produce biofuels through fungal fermentation. This conversion reduces pollution while generating energy. Plastic pollution stands as a major environmental crisis today. Certain fungi exhibit the ability to decompose plastics. Aspergillus tuberculosis can degrade polyurethane. Other fungi are being researched for polyethylene decomposition. These discoveries offer hope for plastic waste management. The construction industry is a major resource consumer. Mycelium composites are transforming this landscape, using agricultural waste as substrate for mycelium cultivation. Produces lightweight, durable building materials. These materials are biodegradable with minimal carbon footprint. The textile industry has traditionally been highly polluting. Fungal leather offers an eco-friendly alternative. Production requires no tanning chemicals. Water usage reduced by 99% compared to animal leather. Emerging as a new favorite in the fashion world. Fungi are indispensable for sustainable agriculture. Mycorrhizal fungi partially replace chemical fertilizers. Trichoderma fungi serve as biological pesticides against diseases. These applications reduce chemical contamination, protect soil health, and enhance crop stress tolerance. Water purification is a new frontier for fungi. Certain fungi absorb heavy metals from water. Other species degrade organic pollutants. This bioremediation technology is cost-effective, particularly suitable for developing countries. Fungi play a pivotal role in forest restoration projects. Inoculating suitable mycorrhizal fungi significantly boosts tree survival rates and accelerates recovery of damaged ecosystems. This is particularly crucial for combating climate change. Fungi offer unique contributions to carbon capture and storage. Mycelial networks sequester carbon in soil, forming stable organic carbon reservoirs. This natural carbon sequestration method proves more economical and sustainable than artificial techniques. Fungi hold vast potential in urban environments. Vertical farms utilize fungi to process organic waste. Edible mushrooms cultivated on building rooftops. These innovations foster greater urban self-sufficiency, reducing reliance on long-distance transportation. Fungi are essential for transforming food systems. Microbial protein production far exceeds livestock efficiency, partially replacing animal protein with fungal protein. Significantly reduces land use, water consumption, and greenhouse gas emissions. Fungal knowledge is becoming widespread in education systems. A new generation is learning to coexist harmoniously with nature. Fungi serve as a window to understanding ecological interconnectedness, cultivating a mindset for sustainable development. Fungi are ideal partners in circular economy models. They transform waste into high-value products. This process generates minimal pollution, achieving truly zero waste production. Fungi offer new possibilities in energy production. Certain fungi enhance biomass conversion efficiency. Other species can be used in microbial fuel cells. Though these technologies are in their early stages, they demonstrate immense potential. In climate change adaptation strategies, fungi help ecosystems maintain resilience. Their mycorrhizal networks assist plants in coping with extreme weather like drought and heat. This natural wisdom is worth emulating. Fungi play a vital role in water-scarce regions. Certain desert fungi absorb moisture directly from the air. This mechanism could inspire new water extraction technologies to address drinking water challenges in arid areas. The integration of traditional knowledge with modern technology is unlocking fungi's greater potential. Ancient indigenous wisdom about fungi complements advanced biotechnology, paving new paths for sustainable development. Economic paradigms transformed by fungi. Biologically based production methods generate green employment opportunities, boost regional economic development, while safeguarding ecological environments. Global collaboration strengthened by fungi. Fungi grow naturally without borders. Scientists studying them cross geographic boundaries to collaborate, jointly addressing global challenges. Policy making turns its attention to fungi. Some nations are integrating fungal conservation into biodiversity strategies. International organizations are promoting equitable use and benefit sharing of fungal resources. Public awareness is rising. More people recognize the importance of these unassuming organisms to Earth's future. Protecting fungi is becoming a shared imperative. Fungi's role in a sustainable future proves a simple truth. Nature holds the answers to all our problems. What we need is to learn to listen and collaborate with these tiny yet magnificent beings. When we survey Earth's web of life, fungi form its most critical connecting threads. These seemingly minute organisms are actually the foundational infrastructure sustaining entire ecosystems. At the microscopic level, fungal mycelium networks resemble the Earth's nervous system, silently transmitting information underground, coordinating interactions among plant communities. This intricate communication system enables forests to function as a superorganism. Fungi are indispensable in material cycles. By decomposing dead organic matter and releasing nutrients locked within. Without fungal decomposition, Earth would be buried under layers of dead organisms, and nutrient cycles would stagnate. Fungi are guardians of biodiversity. Through mycorrhizal networks, they help diverse plants share resources and information. This mutualistic mechanism maintains ecological balance by preventing any single species from overpopulating, thus safeguarding species diversity. Fungi make silent contributions to climate regulation by influencing carbon cycles and moderating atmospheric greenhouse gas levels. Mycelial networks in soil store vast quantities of organic carbon. These carbon reservoirs are vital for stabilizing the global climate. Soil health entirely depends on fungal activity, as they create soil aggregates, enhancing water retention and aeration, while producing humus to improve fertility. Without fungi, soil would lose its vitality. The flourishing of the plant world rests upon fungi. Approximately 90% of terrestrial plants form symbiotic relationships with fungi. This partnership is so successful that it can be said, without fungi, there would be no modern forests. In terms of ecosystem resilience, fungi act as buffers. When the environment undergoes drastic changes, Fungal networks help biological communities adapt to shifts, enhancing the resilience of entire ecosystems. Fungi connect diverse biological groups. They collaborate with bacteria to decompose organic matter, partner with insects to disperse spores, and engage in mutualistic symbiosis with mammals. These extensive connections strengthen the web of life. On an evolutionary scale, fungi drive innovation. Their coevolution with plants shaped the landscape of terrestrial ecosystems. Interactions with other organisms spawned new ecological niches and survival strategies. From mountain peaks to deep-sea hydrothermal vents, from equatorial rainforests to polar tundras, fungi inhabit nearly every habitat. This widespread distribution demonstrates their exceptional adaptability to Earth's environments. Fungi embody the wisdom of life. They do not win through speed or strength, but through patient connection and transformation. This philosophy of survival may be precisely the ecological wisdom humanity needs to learn. When contemplating biodiversity conservation, we must not focus solely on visible plants and animals. We must also protect these subterranean engineers, for they are the true cornerstone of ecosystems. Every time we disrupt fungal networks in the soil, we undermine the foundation of the web of life. Every time we protect a forest, we also safeguard these invisible connectors. Each new scientific discovery reinforces our understanding of fungi's importance. The influence of these microscopic organisms is closing the gap with our comprehension. Educational systems must teach more about fungal ecology, empowering the next generation to grasp their pivotal role in sustaining Earth's life. Policymaking should prioritize fungal conservation, incorporating fungal diversity into conservation goals, establishing fungal resource reserves, and developing sustainable utilization standards. The business community is recognizing fungal value, from environmental protection to product development. These organisms offer boundless commercial opportunities, while also bringing responsibility. The general public can protect local fungal diversity through simple actions, such as reducing chemical pesticide use and safeguarding natural habitats. Global cooperation is vital for fungal conservation, as these organisms transcend national borders, requiring collective international effort to effectively protect their diversity. Reflecting on Earth's evolutionary journey, fungi have always been silent witnesses, yet also active participants. They shaped the Earth of the past and will influence the world of the future. In the Anthropocene, this new geological epoch, we need fungal wisdom more than ever. They teach us how to coexist harmoniously with nature, how to build sustainable relationships. When we finally comprehend the indispensable role of fungi in Earth's web of life, we will realize that protecting them is in essence protecting ourselves and the world we all depend upon. This understanding will guide us toward a wiser future where humanity, fungi, and all other life forms weave a more beautiful web of life together. Personal lifestyles will undergo profound changes. People cultivate edible fungi at home, utilize fungi to decompose kitchen waste, select eco-friendly products sourced from fungi. Daily life becomes intimately connected with nature. Spiritual dimensions gain new insights. Fungal growth patterns teach us the importance of patience, persistence, and interconnectedness. Their survival wisdom reminds us true strength stems from cooperation, not competition. Technological development follows ecological principles. Internet design mimics mycelium networks. Fungal decomposition inspires eco-friendly technologies. These bio-inspired innovations enable technological progress to coexist harmoniously with the environment. Environmental protection will be grounded in a new philosophy, no longer viewing nature as an object to conquer, but as a partner. Humans, like fungi, will become builders and maintainers of ecosystems. This vision of harmony is not a distant dream, but a reasonable extrapolation from existing scientific understanding. Numerous successful cases worldwide demonstrate the feasibility of human-fungal symbiosis. Realizing this vision requires our collective effort. Scientists must deepen fungal research. Entrepreneurs must invest in fungal tech innovation. Policymakers must create enabling environments. Citizens must transform consumption habits. Educational institutions must cultivate interdisciplinary talent. Artists must create inspirational works. Media must disseminate accurate scientific knowledge. Everyone can play a role in this transformation. Looking ahead, we envision not a world where humans dominate nature, but one where diverse life forms coexist harmoniously. Humans, as part of the ecosystem, will establish equal and mutually beneficial partnerships with other organisms, especially fungi. This symbiotic relationship will help humanity resolve current environmental crises and achieve true sustainable development while enriching our lives and making our civilization wiser. When our great-grandchildren ask how humanity found the path to sustainability, we can tell them, we learned from Earth's oldest inhabitants how to coexist harmoniously with all life. This may be the most precious gift fungi bestow upon humanity, not a specific technology, but an entirely new way of thinking, a wisdom for living in harmony with nature. This wisdom will guide us toward a brighter and more sustainable future. In this future, humanity will finally understand we are not nature's rulers, but a single thread in the web of life, together with fungi and all other beings, weaving the beautiful tapestry of Earth's life. Let us embrace this new era of symbiosis with fungi, with reverence and gratitude. In this age, human civilization will achieve true harmony and unity with nature, jointly creating a vibrant Earth home. As an ancient proverb states, when we truly comprehend the value of small things, we gain the power to change the world. These tiny fungal lives are guiding us toward a brighter tomorrow.